Here, in this state-of-the-art facility, interleukin scientists are hard at work developing new genetic tests, the results of which may very well change the course of your health someday.
We use this cryogenic tank to preserve living cells. Here in the Research and Development Lab, we study these cells to better understand how DNA works. Through this kind of research, we can develop new products and new genetic tests for additional pre-existing conditions and diseases.
Our laboratory passed the government's Clinical Laboratory Improvement Amendments to ensure the highest quality of privacy, accuracy, and quality. Within this lab, our highly experienced scientists work with computer-driven robotics and analysis equipment to deliver reliable results for every single test. Let's quickly go through what happens to your sample after you send it to Interleukin Genetics. First, the test swab arrives at our facility. Your information is logged into our laboratory information management system. The system barcodes the sample and tracks the barcode throughout the testing and analysis process. Then, samples are passed through a one-way airlock into a clean room, a work area with highly controlled temperature, humidity, and airflow. The clean room protects your swab and DNA from contamination. Not all genetic testing labs have a clean room, but having a clean room leads to a more reliable test result.
We take someone's DNA from the swab and test it for certain genetic variations. We first make specific primers, which are tiny pieces of single-stranded DNA that are specific to the area we want to target. Then we use computer programs to search the entire human genome to ensure that we're targeting the correct piece of DNA. Next, we do PCR, or polymerase chain reaction, a technique that amplifies a section of DNA and causes a chain reaction to keep amplifying the DNA, giving us millions of copies to visualize.
Next, we use a technology called single-base extension. This highly accurate method is used to identify known single nucleotide polymorphisms, or SNPs for short. SNPs are variations in your DNA sequence that may help identify risks for various diseases and conditions. The single-base extension technology labels SNPs with a fluorescent dye. This dye is then visualized by this very specialized machine behind me. The results from this instrument are used to determine your individual genotypes and any predisposition to the disease they may indicate.
This information then goes back to the Laboratory Information Management System, where a test report is created.
For more than 10 years, Interleukin Genetics has been focused on understanding how gene variations that affect inflammation alter your risk for certain diseases. There is no other test like the Gensona genetic test. Interleukin Genetics has worked hard to earn patents on the gene variations for these tests. The Gensona genetic test provides information never before available, so you can take steps to lead a long and healthy life.